Jay has come from the Beaton in Glasgow, and he is going to talk about, if I can read properly, pancreatic cancer tissue architecture using spatial omics. So still in the spatial scene, thank you.
Hello everyone, I'm Jaya from CRUK Scotland Institute. I'm working on mapping and modeling pancreatic cancer using spatial omics and agent-based mathematical modeling. So you may know pancreatic cancer is one of the most aggressive cancer types. The 10-year survival rate in England is about 5%, but still many cases, like 1/3 of the cases, are preventable. Therefore, of course, we need some novel strategies to better understand pancreatic cancer. So that was the motivation for my project. And then more than 80% of pancreatic cancer cases are ductal type. We commonly call them PDAC. And PDAC is characterized by a dense fibrotic stroma, which can account for up to 90% of the tumor volume. And the major contributor for this stroma is the cancer-associated fibroblasts. So in PDAC, CAF can originate from different kinds of cells, pancreatic stellar cells, fibroblasts, immune cells, etc. And they can differentiate into different subtypes. The major subtypes are MyCAF, ICAF, and antigen-presenting CAF, and they have different kind of roles within the PDAC. And then the overall research questions for my project are this. I need to understand how the phenotypic subgroups of CAFs are organized in a spatial context by using spatial transcriptomics data. And also I need to understand how CAF phenotypic and spatial heterogeneity emerge mechanistically by using an agent-based mathematical model. So first I analyze some spatial transcriptomics data from this recently published paper. The data set contained untreated patients and also patients treated with chemotherapy and radiotherapy. Then individual tissue samples contain several FOVs, field of views, and inside those FOVs I have spatial transcriptomics data for a targeted panel of 1,000 genes. Then I ask the spatial distribution of MyCAF, ICAF with respect to cancer cells. To do that, I calculate the proximity score for MyCAF and ICAF. In this case, larger the proximity score means more CAFs are within cancer cells. So we can see the MyCAF proximity score is higher than the ICAF proximity score, indicating that across all untreated samples, MyCAFs are enriched near cancer cells. So then I develop my mathematical model to understand crosstalk between cancer cells and CAF, and also CAF differentiation into MyCAF and ICAF subtypes. So from literature we know CAF near to cancer cells differentiated to MyCAF through TGF-beta signaling molecules secreted by cancer cells, and also CAF located farther away from cancer cells can differentiate into ICAF phenotype through IL-1 signaling molecules secreted by cancer cells. Then I implemented these gene signaling pathways in a Boolean network and integrated into a multi-scale agent-based mathematical model to simulate individual FOVs within tissue samples. So I initialized my mathematical model with precise locations of cancer cells and CAF, and then I asked my mathematical model CAF subtypes in space and time. So this is the model output. Then I compared my model predictions with spatial data analysis. We can see that depending on the tissue architecture, we get different proportions of ICAF and MyCAF, indicating that tissue architecture may impact on CAF phenotypic evolution. So if you are interested in knowing more about my work, so please visit my poster. Thank you.